dear students. In this session, we are going to discuss the bacteria Streptococcus, which is a very important bacteria. After we have learned Staphylococcus, which was gram-positive cocci, which is seen in groups. This one, Strepto, means it is seen in chains. Staphylo means it was bundled. Strepto means in chains. So Streptococcus is a gram-positive cocci. From the name itself, you can see it is a cocci and it is seen in chains. So the most important questions from this topic is, which was previously asked, antigens, enzymes and toxins of Streptococci. Actually, all these are the virulence factors of this bacteria, which is a very important question. And you also have to learn what are the diseases caused by Streptococci and how will you diagnose the disease. And we have a classification for Streptococci that can also be a probable question which can be asked for exams. So this is the classification. So in this image, you are seeing cocci in chains. You can see that. What happens actually is when this cocci divides and becomes two, it won't get separated from each other. So bacteria divides by binary fission, but it fails to get separated from each other. That leads to appearance as chains in microscopy. So gram-positive cocci arranged in chains, that is how we describe the morphology of Streptococci. And how we can classify this group of bacteria. Basically, we can classify Streptococci based on their oxygen requirement. When we simply say Streptococci, we actually mean the aerobic group of Streptococci which require oxygen for growth. But there are anaerobic Streptococci which do not require oxygen for growth. They are called as peptostreptococci. So peptostreptococci are anaerobic Streptococci, and whereas when we simply say Streptococci, we mean the aerobic ones, that is the normal Streptococci. Now, based on how they grow on blood agar, in blood agar, what is the most common property that we can elicit from blood agar is whether the bacteria is lytic or not. Most of the bacteria, like Streptococci and Staphylococci, they can produce certain hemolytic toxins which can break down the RBC so that in blood agar they look hemolytic, they produce lysis of the RBCs. So what kind of lysis they produce on blood agar, based on that you can classify Streptococci. So if they are producing an incomplete hemolysis, it will produce a greenish color on blood agar. I'll show you in the next image. Those are called as alpha hemolytic Streptococci. So please remember, alpha hemolytic Streptococci means it is incomplete hemolysis, and you will see a greenish color discoloration because there is no complete lysis. It will be just a partial lysis. So the best examples for alpha hemolytic Streptococci are Streptococcus viridans. Viridans is actually a normal flora of the oral cavity, but sometimes it can be pathogenic. It can cause certain conditions, but most commonly they are normal flora, Streptococcus viridans. And the second one is Streptococcus pneumonia. Streptococcus pneumonia, it is commonly called as pneumococci. So pneumococci is nothing but Streptococci pneumonia, and both Streptococcus viridans and Streptococcus pneumonia are alpha hemolytic Streptococci. Now coming to the next most important one, that is beta hemolytic. That means in blood agar, you will see this is the colony of the bacteria. You will see complete zone of hemolysis around the colony. There will be complete clearing of the blood around the colony. That is called beta hemolysis. It is complete hemolysis. So under which come most of the important Streptococci, including Streptococcus pyogenes, which we are going to learn in this session. So that is Streptococcus pyogenes. So complete hemolysis is produced by which Streptococci? That is called as Streptococcus pyogenes. See, the name itself say pyogenes, that means it is pus forming. That is why it's called as pyogenes. So this Streptococcus pyogenes has got certain cell wall antigens. Based on these cell wall antigen called carbohydrate antigen, C carbohydrate antigen on the cell wall, you classify the Streptococcus pyogenes into different groups, like it is around 21 groups, and this classification is called as Lancefield classification. So Lancefield classification is mainly for Streptococcus, which shows complete hemolysis. Streptococcus pyogenes is just one example for a beta hemolytic Streptococci, and most of these beta hemolytic Streptococci has got some C carbohydrate antigen on its cell wall. Based on that, you put them into different groups, and that grouping is called Lancefield grouping. So Lancefield grouping, you divide them into different groups from A to U, and this group A is actually our Streptococcus pyogenes. So Streptococcus pyogenes belongs to group A of Lancefield's classification. Actually, there are different other groups, and another important group is group B, which includes Streptococcus Agalactiae. Agalactiae is actually seen as a commensal over the female genital tract, and it can cause occasionally neonatal sepsis. It can affect the young neonates when during delivery they can acquire the infection from the mother's genital tract, and it can lead to neonatal sepsis. That is Streptococcus agalactiae, which comes under Group B of Lancefield classification. So Streptococcus pyogenes is Group A of Lancefield classification. So all these are the different classification of beta hemolytic streptococci. Remember, it is based on the C carbohydrate antigen, and Group A of Lancefield grouping includes Streptococcus pyogenes, and Group B is Streptococcus agalactiae. Now Group A is again classified based on the M protein. There are three proteins, M, T and R, of which the most important one is M protein. Based on the M protein, differences in the M protein, you classify Group A of Streptococcus pyogenes into more than 100 types, and this typing or classification is called as the Griffith typing. So Griffith typing is for Group A Streptococcus alone based on the M protein into more than 100 different types or groups. So Lancefield grouping, you put it under different groups. Group A Streptococcus pyogenes, Group B Streptococcus agalactiae. That classification is based on the C antigen, and again Group A is divided into 100 types based on the M protein, that is called as the Griffith typing. Now there is another group of streptococci which doesn't show any kind of hemolysis. Mostly it can be non-hemolytic streptococci is what we refer to as the gamma hemolytic streptococci, and rarely they may show some degree of alpha or beta. It's variable hemolysis. And the best example for a gamma hemolysis group is Enterococcus. Enterococcus, it's a kind of streptococci which is seen as pairs and short chains, and they can cause wound infection, urinary tract infection, and various other infections. So that is about the different classification of streptococci. And here about the Streptococcus pneumonia, no, it also appear as oval cocci in pairs. It's actually like this. They will be appearing as pairs, flame-shaped cocci in pairs. So mostly streptococci is seen in chains, including Virulence, Streptococcus pyogenes, Streptococcus agalactiae, and even to some extent Enterococcus. But when you say about Streptococcus pneumonia or pneumococci, they appear as pairs, and you will learn it in a separate session. So this is the classification of streptococci. You can see the partial hemolysis one here. Can you see the greenish color discoloration of blood agar? This is because of the partial hemolysis, and the best examples are Streptococcus virulens and pneumococci. And remember, pneumococci, it is seen as pairs in staining, and virulens is seen as chains. It will be seen as long chains. So this is the partial hemolysis or alpha hemolytic streptococci. Now coming to beta, you can see the complete zone of hemolysis in beta. There is complete clearing around the colonies. Examples are Streptococcus pyogenes and Streptococcus agalactiae. Remember, pyogenes belongs to Group A of Lancefield grouping, and agalactiae belongs to Group B of Lancefield's grouping. Both of them are beta hemolytic. Gamma hemolytic or non-hemolytic streptococci do not produce any hemolysis. Enterococcus species, you can see there is no hemolysis in the blood agar. So this is the classification. It can be asked as a question. Now coming to the morphology in detail of Streptococcus pyogenes. So we are going to learn in detail only Streptococcus pyogenes in this session. That is Group A Streptococcus. They are spherical or oval in shape. Their diameter, you can see it's a very small one, 0.5 to 1 micrometers. They are gram-positive cocci in chains. You can see the cocci in chains here. Non-motile, non-sporing. Most of the cocci are non-motile and non-sporing. Now their requirement for oxygen, I told you they are aerobes and facultative anaerobes. When we say about obligate anaerobic streptococci, that is peptostreptococci, that's a different entity. This one grows best at the normal temperature of human body, that is 37 degrees Celsius. They can grow best in medium containing blood and serum. They cannot grow on nutrient agar. They are fastidious. They require blood or serum for growth. And growth and hemolysis can be promoted if you provide them with some carbon dioxide. If you give 10% carbon dioxide during incubation, you can make the growth better. Now coming to the most important question from this part, this was asked repeatedly, what is the antigens, toxins and enzymes produced by streptococci? All these are actually the virulence factors. They contribute to the pathogenesis of this bacteria, virulence factors. So virulence factors of streptococci can be classified as the structural antigens, the toxins that they produce, and also the enzymes. So these three are the three important virulence factors of streptococci. So coming to the structural antigens or the antigenic structure, this is very important. You can remember that with the code CPM. So the structure is constituted by CPM, C for capsule, P for polysaccharide antigen, and M for M protein. So capsule is actually composed of hyaluronic acid capsule, and all the strains doesn't have this one. Only few strains will have the capsule. It is composed of hyaluronic acid. Now when you see the cell membrane, you can see inside you have the cytoplasmic membrane. So this is the cytoplasmic membrane. And outside the cytoplasmic membrane, actually this layer is composed of the peptidoglycan, which is present in all gram-positive bacteria. So that is the peptidoglycan layer, which is present for all gram-positive bacteria. Now outside the peptidoglycan layer, there is a group-specific polysaccharide or carbohydrate antigen layer. So you call that as C layer, C antigen or C carbohydrate antigen layer. That is what the polysaccharide antigen means. So that is just outside the peptidoglycan. And again, when you see the next layer, it is composed of protein and lipoteichoic acid. It is from this layer that the fimbriae of this bacteria arises from this protein and lipoteichoic acid layer. So this protein layer, it is composed of different proteins like M, T and R, of which M is the most important one. So these are the different structural antigens of streptococci. CPM, capsular hyaluronic acid, P for polysaccharide or carbohydrate antigen, and M for M protein. M protein is the most important one. It has got different other proteins also. So the pili or the fimbriae, it is covered with the lipoteichoic acid and also the proteins because it arises from that layer. So this is the structural antigenic layer of streptococci. And actually this layers has got some importance. Why? Because these layers, these antigens mimic certain human antigens. For example, this capsular hyaluronic acid layer, it has, because it is composed of hyaluronic acid, it has some cross reaction with the antigens present in human joints and synovium. So antibodies produced against this capsular hyaluronic acid can cross react with the human joints and it can cause some pathogenesis. Similarly, the polysaccharide or the C antigen layer, it has got cross reaction with the cardiac valves in humans, human cardiac valves. Similarly, the M protein has got some cross reaction with the myocardium of humans. So the capsular layer, which is composed of hyaluronic acid, you can remember the synovium is also composed of hyaluronic acid. So they have cross reaction. The C carbohydrate antigen, C for C cardiac valves, and M protein, M for M myocardium. So since they are having cross reaction with all these human tissues, antibodies produced against these three antigens can cross react with the human antigens and it can cause tissue damage and therefore it can lead to certain complications in streptococcal infections. So what is the role of capsular hyaluronic acid? Any bacteria which has got a capsule, it will inhibit phagocytosis. So that allows the bacteria to survive the human immune cells. So it will inhibit the phagocytosis. And when we say the cell membrane layer, as we have already seen, the outer layer, protein and lipoteichoic acid from which the fimbriae arise. The middle layer is composed of the group specific C carbohydrate antigen and the inner layer is composed of the peptidoglycan layer. Actually, this peptidoglycan layer has got some cross reaction with the human skin antigens. C carbohydrate antigen with the cardiac valves. I'm repeating this because this is the reason for the pathogenesis of the complications in streptococcus like rheumatic fever. And the protein layer or M protein layer with myocardium. So these are the cross reactions. So you call the C carbohydrate antigen as the group specific polysaccharide antigen. So these are the important structural antigens. Now, when you say about the type-specific antigen, we mean the proteins, the M protein, T and R protein. Actually, M protein also helps in inhibiting phagocytosis, not just capsule. And it is based on M protein that we classify Streptococcus pyogenes or group A Streptococcus into different types, and that different type is called the Griffith typing. Right? So Griffith typing is based on the M protein. This is purely for group A Streptococci. Group A Streptococci is further classified based on the M protein into different Griffith types. And I hope you remember what is the Landsfield classification. Landsfield classification is based on the C antigen or carbohydrate antigen, for which you are classifying them into A to U groups. And our group A Streptococcus comes under this group A. A is for group Streptococcus pyogenes and B is Streptococcus agalactiae. So just a revision on the classification. So we have finished the antigens of Streptococci. The next thing is toxins. What all toxin does it produce? So you can remember that with a mnemonic SP. S for streptolysin and P for pyrogenic exotoxin. S for streptolysin. Streptolysin is actually hemolysin produced by Streptococci, which has got two types, O and S. Streptolysin O means it is oxygen labile. S means it is stable. S for stable and O is labile. So there are two hemolysins produced by Streptococci. So since it is produced by Streptococci, you call them as streptolysin. So what do? They lyse the red cells. They are cytotoxic for neutrophils and platelets, and they are highly antigenic. Streptolysin O is antigenic. So if we have an infection with Streptococcus, our body produces anti-streptolysin O antibodies. You call that as ASO, anti-streptolysin O. Now, what is the role of streptolysin S? It is oxygen stable, so that is responsible. If you have a blood agar plate with the colonies, you get a lysis around the colonies. This is because it is producing streptolysin S. Since it is oxygen stable, it can cause lysis even on the surface of agar. So that is responsible for the lysis around the colonies on blood agar, and they are not antigenic. They don't elicit the production of antibodies. So antibodies are produced only against streptolysin O. So you can detect this ASO antibodies for diagnosis of streptococcal infection. So these two are the hemolysins produced by Streptococci, streptolysin O and S. And the next toxin, SP, pyrogenic exotoxin. It's also called as erythrogenic toxin. So from the name, you can understand pyrogenic means it will produce fever. Pyrogenic means fever producing. Erythrogenic means it will produce erythematous rashes. So they are responsible for the rash of scarlet fever, and they are produced by certain lysogenic strains. Lysogeny means this pyrogenic exotoxin, the property of producing this toxin is coded to the bacteria by certain phages. That is why it is seen only in the lysogenic strains. So pyrogenic exotoxin is responsible for the fever and rash of scarlet fever. There are two tests based on the pyrogenic exotoxin. Actually, both these tests are outdated and not performed now, but still we learned this in the theory part, or you can get an MCQ from this. Dick test and Schulz-Charlton reaction, both these are based on the pyrogenic exotoxin. So what we do, if you inject some toxin, pyrogenic exotoxin, intradermally into the skin of a susceptible child who is susceptible for scarlet fever, you can see at the site of injection, there is a localized erythematous reaction. If this reaction is positive, you consider Dick test as positive, and that indicates the child is susceptible to streptococcal scarlet fever. Now, suppose if a child is already having scarlet fever, we suspect scarlet fever. If you inject the homologous antitoxin to the pyrogenic exotoxin, you are injecting the antibodies to the pyrogenic exotoxin into the rash, which is produced in scarlet fever. You can see there is blanching of rash because this is because of the toxin-antitoxin interaction or reaction, that there is blanching of a rash when you inject antitoxin into a rash. So that is called as Schulz-Charlton reaction. At least remember Dick test and Schulz-Charlton reaction, it is based on pyrogenic exotoxins and it is related to scarlet fever. Coming to the last part of the antigens, enzymes and toxins. Now that is what the virulence factors are composed of. So we have finished antigens, we have finished toxins. The next one is the enzymes produced by Streptococci. These enzymes are very important in the pathogenesis of Streptococci, which helps in the spread of infection into the planes, into the subcutaneous tissue, muscles and all. So there are different enzymes. The most important one you can remember with the code DHS. D for DNase, H for hyaluronidase and S for streptokinase. So streptokinase, DNase, hyaluronidase are the most important one. Also we have NADase, nicotinamide adenine dinucleotidase is also there. That is called NADase. It is less important. Remember the other three, that is more important. What is the role of each of them? Streptokinase. Streptokinase is nothing but it's a fibrinolysin. So what does fibrinolysin do? It will lyse the clot. No? So clot is composed of human fibrin. If this fibrinolysin lyses the clot by catalyzing conversion of plasminogen into plasmin. So when plasminogen is converted to plasmin, it will cause lysis of the fibrin clot. So why it is important? When there is an infection, when there is an inflammation, there can be induction of the clotting factors and some clot will be formed which will be trying to contain the infection. But this streptokinase will lyse this clot and it is helping the bacteria to spread further. So this one is protein in nature, it is antigenic, it will elicit the production of antibodies, and this facilitates the spread of infection. So streptokinase facilitates the spread of infection by breaking down fibrin barrier around the lesions. That is what I told you. If a bacteria is multiplying like this, sometimes a clot will be formed, a fibrin clot will be formed which tries to contain the bacteria. So this bacteria, it will lyse this fibrin clot and it will spread further. So it helps in spreading of the infection. Now, what is the role of DNAs? DNAs will break down the deoxyribonucleic acid. There are 4 types of DNAs and B is the most antigenic one. And when it liquefies this DNA, what happens? No, the pus will be liquid, very serous in nature. It won't be purulent. It will be watery or serous in nature because the nucleic acids are broken down. So this is responsible for the serous nature of pus in streptococcal infection. The pus is not thick, it's very watery or serous. Coming to hyaluronidase, it breaks down the hyaluronic acid of the tissues. This also, so the cartilage, everything will be composed of hyaluronic acid. No? So if it breaks down the hyaluronic acid, this also helps in spreading of infection along the intracellular spaces. So all the 3 enzymes, streptokinase, DNase and hyaluronidase, helps in spreading of the infection. DNase also help in liquefying the pus and making it serous in nature. So that is about the virulence factors, and we'll just see what are the diseases caused by Streptococcus pyogenes. So we are talking about Streptococcus pyogenes. Please remember, I'm repeating it. This is group A Streptococci. So it can cause skin infections and deep infections. So what are the skin infections? It can produce impetigo. It will be crusted lesions over the skin, especially it is seen in small children. And erysipelas is a localized form of cellulitis, which is commonly seen over the face. Cellulitis means it is inflammation of the skin and the subcutaneous tissue. It can affect any part, particularly over the limbs. Necrotizing fasciitis, that is a more deeper infection produced by streptococci. It will be spreading from the skin to the subcutaneous tissue, to the fascia, and sometimes it can reach up to the muscles. So it is going deeper and deeper. That is what is happening in necrotizing fasciitis. So there will be complete necrosis of the skin and soft tissues up to the fascia and muscles. It will go. Now it can cause certain upper respiratory infections. So when somebody asks you what is the most common cause of pharyngitis and tonsillitis in a young child, the most common cause is Streptococcus pyogenes, bacterial cause. Okay, the other causes can be viruses, but among bacteria, the most common cause of tonsillitis and pharyngitis is Streptococcus pyogenes. And in addition to this, in any kind of infection, particularly when a Streptococcus pyogenes cause cellulitis and necrotizing fasciitis, it can further enter the bloodstream. It can cause bacteremia and septicemia. So, following necrotizing fasciitis, bacteremia and septicemia is very common with Streptococcus pyogenes if you don't treat the patient. Now, in addition to all these suppurative infections, why all these are called suppurative infections? Because there is pus formation in all these infections. That is why we call it a suppuration. There are non-superative complications, that is rheumatic fever and acute glomerular nephritis. You know the reason? We have already discussed the reason. This is because of the cross reaction between streptococcal antigens and human tissues. So that is what I told you. The cardiac valves and C antigens are similar. Myocardium and M antigens are similar. The hyaluronic acid and joint antigens are similar. Peptidoglycan and skin antigens are similar. So the cross reaction can lead to different lesions in all these areas. It can lead to endocarditis, myocarditis, joint involvement, synovial effusion and synovitis, joint effusion, skin lesions like rashes and nodules. All these occurs in rheumatic fever. This is because of the cross-reacting antigens. Similarly, acute glomerulonephritis, we have learned it in hypersensitivity. I hope you remember it is a type 3 kind of hypersensitivity because of deposition of immune complexes. So immune complexes get deposited over the glomerular basement membrane because there is some similarity between the cytoplasmic membrane of Streptococcus and the glomerular endothelium and basement membrane. So the immune complex will go and get deposited there. That can also elicit inflammation leading to nephritis. So please remember what are the complications of streptococcal infection, rheumatic fever and acute glomerulonephritis. Finally, coming to the laboratory diagnosis, we have various specimens that we collect for Streptococcus. You can imagine the specimen. If suppose the child is having upper respiratory tonsillitis or pharyngitis, your sample will be throat swab. If they are having necrotizing fasciitis, cellulitis, erysipelas and all, your sample will be pus or tissue. If it is a septicemia case, it will be blood, and very rarely if it is a meningitis, it can be CSF. but it's very less common with Streptococcus pyogenes. And how will you send the sample? If there is a delay in sending the sample to lab, please use a transport medium that is called Pike's transport medium, because this bacteria is very delicate, fastidious. It will die easily, so you have to send it in a transport medium. And in lab, what you're going to do, which culture medium we're going to use? We are going to use a blood agar. That is the best medium for Streptococcus. You incubate at 37 degrees Celsius for a day, and you will see hemolysis. What kind of hemolysis is produced by group A Streptococcus? Beta hemolysis or complete hemolysis. So this can be produced better under anaerobic conditions or by providing 5 to 10% carbon dioxide. So if you provide carbon dioxide, the hemolysis will be very beautiful. And you can use a selective medium for Streptococcus pyogenes if you want to differentiate it from other hemolytic bacteria. It is called crystal violet blood agar. So that is the selective medium that we use. So initially, before culture, what will you do with the samples at first? Always your first point should be gram staining. So in gram staining, you're going to see gram-positive cocci in long chains. So directly you can do gram staining, and also from the colonies you can perform gram staining. So colonies will be beta hemolytic, and when you perform gram staining from the colonies, this is how it looks like, long chains of gram-positive cocci. And the other important test are this bacteria is catalase negative. I hope you remember what is the catalase positive GPC, gram-positive cocci, which is catalase positive. That is nothing but staphylococci. So catalase is the basic test which differentiates staphylococci from streptococci. And the other test which is positive for Streptococcus pyogenes, it is bacitracin sensitive. This is an antibiotic, bacitracin. If you put a disc of bacitracin, there will be no growth around this disc. The bacteria will be inhibited by bacitracin. So that is a specific test for Streptococcus pyogenes. All other streptococci are bacitracin resistant. Finally, treatment. What treatment you can give? The best and the most widely used drug for Streptococcus pyogenes is penicillin G. Penicillin is not at all found resistant for streptococci, so we can directly give penicillin for any case when you're suspecting Streptococcus pyogenes infection. Only if the patient is allergic to penicillin, you can give erythromycin. So prophylaxis, if a case of rheumatic fever, if you want to prevent, it's very important to treat streptococcal sore throat, because the complications of rheumatic fever and glomerulonephritis, especially in young school-going children, this is very common. So you have to give penicillin, and if suppose they develop rheumatic fever, you have to give a long-term penicillin therapy for preventing further complications, and that is they can lead to permanent cardiac complications. So to prevent reinfection and damage to heart, you have to give a long-term penicillin therapy if you have to prevent future damage from the bacteria. So once more, I'm repeating the important question: antigens, toxins, and enzymes. Also, please learn what are the diseases caused and how will you diagnose, lab diagnosis of streptococci. And don't forget, this is gram-positive cocci which appear in chains. The morphology is important, and the only drug that you have to remember for streptococci is penicillin. So please learn the structure. That is important. That is the most important part of this topic. Thank you.